It's a real pleasure to be here today and to be back in person for a biophysics meeting. Thank you so much to the organisers for putting this on. It's been absolutely brilliant. So today I'm going to talk to you a little bit about DNA, but going back a little bit from where we've just been thinking about DNA replication and going really back to the structure of DNA and what we do and don't know about that structure. I think in a biophysics community, it's quite safe to say that we understand the importance of dynamics, heterogeneity, the role of sequence and structure in interactions. But I think it's always nice to go back to when we think about the double helix of DNA, thinking about how many different conformations and shapes it can take up and how that recognition can change how it interacts with things. And one of the things I always like to show is this crystal structure of IHF complex with DNA. And it's a really lovely paper that shows that DNA can make a full U-turn around an IHF protein. But actually, this was only obtained by annealing a load of oligos together and leaving some big gaps in order to allow this to crystallize. And I think we know that every experiment we do has flaws. But there are questions there which really speak to me, which say, is it the gaps that allow the protein to sit there, that allow the crystal to form? Or is it the gaps that allow the DNA to pack? And you get into a chicken and egg conversation saying, what came first, the defects or the binding? What came first, the interaction or the changes to the structure? And we are really interested in how the structure of DNA affects all of its interactions. So as you might have guessed from my title, I bolted on my technique to my title to make it very clear. I'm an atomic force microscopist by training. But AFM has some really amazing reasons for looking at DNA, or some attributes for looking at DNA. So if we look at here, what we do is we read out a surface using a large cantilever, which acts like a record player needle or a finger reading out a piece of braille. And the things that are important to take in this image are the fact that there's a blue bubble on top of your sample, which means we're working in liquid. And also the fact that we're interacting physically with the sample. So it's kind of good and bad on that front. And so what we're able to do is actually read out each individual molecule and look at the structure of those molecules by feeling them. And there's a bit of a caveat there that we have to stick everything down. And we don't like to talk about that very much, but it is a very essential part of what we do. And it's sort of important to put that out there up front. It's not just lying there, it is slightly blue tacked down. And when we use AFM, we're actually able to see the structure of DNA. And we're not the first people to do this. This started a long time before me. And I think when we're talking about next generation biophysics, it's also important to talk about what came first, right? So this is an image from Carlos Bustamonte's lab where they first imaged DNA in air a long time ago. And actually people back as far as the mid-90s were able to image submolecular structure of DNA by closely packing it together on lipid bilayers and by stopping it being able to move around. So a bit more super glue to our blue tack. And moving on, it was quite a few years before there was much change to the way we were able to see DNA using AFM. There was a lot of work put into improving the resolution of AFMs and the surface chemistry which we used to stick these down. But actually, it wasn't until 2012 when two papers came out, one from my PhD supervisor, Bart Hoevenboom's lab that I was involved in, and one from a group Yamada's lab in Japan came out at exactly the same time, showing submolecular imaging of DNA, so showing the double helical structure, both in left and right-handed DNA, showing the major and minor groove. And this was a huge step forwards for what AFM was able to do. And that was amazing, apart from the fact that these were both done on home-built systems that only existed in 2 labs in the world. And anyone in this lab that's used home-built systems for very complex biochemistry work knows that the likelihood of those 2 working together at the exact same time can be quite low. So we did a lot of work here with a manufacturer trying to move some of the amazing step forwards that had been made in instrumentation onto commercial equipment so we could start improving the throughput of our experiments and actually trying to interrogate biological samples. And that's what happened quite recently. And so we were able to see the double helix of DNA in situ. And I've showed this image before, but I like to start with it because I think it gives you an idea of where we're coming from. So we were imaging plasmids of DNA, and when we zoom in, we're able to see the major and minor groove of DNA. And what I've done is blow up this piece here and overlay the B form of DNA, just made a bit longer. And the reason I've done that is not because I think we're crystallographers or because I've got ideas above my station, but it's to show you what we can and can't see. So everything in sepia is usually AFM, and the rest of the colorful things are not. So here you can see that in the crystal structure, we see the bases, and in AFM, we do not. You can see, though, that we can see the major and the minor and the major and the minor and the major, minor and major grooves of DNA. But what you can also hopefully see is that there's quite a lot of variation there. So we've got areas where we've got very large major grooves of DNA and areas where those are barely visible at all. So we're seeing heterogeneity along a single molecule of DNA in a small sort of 20 nanometer window. We're seeing changes in that helical structure that we can only see by directly looking at one molecule. We lose this when we start to average. So that's the great thing. We can see variation in a single molecule of DNA, and we can see the helical structure, and we can resolve back the correct pitch and things like that. But the things you might have noticed are this is quite a lot wider than the crystal structure. The DNA isn't suddenly 5 nanometers wide. We have an issue with our technique where we're physically probing it and we get a thing called convolution. In an AFM, like the surfaces, we tend to ignore that. We showed that we can reconstruct that out, but it's quite computationally expensive and we tend not to do it. But one of the artifacts that that gives is this actually changes the chirality of the helix. So this is quite vertical in shape compared to the chiral angle here. And that's an artifact of this broadening. But we can again reconstruct that out and we get the correct helical angle, we get the correct width, we get the correct height. So everything matches nicely, but we see this variation. So what can we use the fact that we can see this structure to find out? And how did we get there? Well, I talk about this as being the spaghetti to fusilli reaction, change in resolution, where we suddenly go from seeing DNA as worm-like change with no features to being sort of structurally relevant. But actually what this involved was 4 years of silicon microfabrication in the clean room, because it turns out that the rate limiting step here was the cantilevers that were being fabricated that were designed to have end radii between 1 and 20 nanometers. Now, as a single molecule person, that kind of stretch around a 2 nanometer piece of DNA makes me anxious. And so what we did was we worked with Brooker for about 4 years to make cantilevers that were soft enough, so as softer as a piece of tinsel, to give you an idea, to be able to move over the DNA and give us this resolution without crushing it. Because for size, this tip here is about the same size as a large sort of Matterhorn-esque mountain, if the DNA is about the same size as a football. So you're floating a mountain over a football, and you've got to get that mountain to be football sharp at the end. But we've done this, and these exist now, and you can buy them. So we can now do this reproducibly, and we can start looking at things on a much better scale and getting ends that are not quite so bad. And we can start looking at different populations of structure in DNA. So now we can do that. What can we do to actually start looking at DNA structure? Well, the first thing we did was make our system smaller and more discrete, because plasmids are actually quite big on an AFM scale. They're quite flexible. I can get into convoluted messages about headphones and how things get entangled. But really what we did was make small mini circles, twice the persistence length of DNA. And the reason for that is basically a discrete entity. You've got a length over which the DNA basically wants to be straight. So you create a planar molecule. And if you're going to put something down on a surface, making it planar first makes that a lot more straightforward. It also had the added advantage that our wonderful friends could simulate these in all atom MD simulations in explicit solving. And the first thing that I love here is how well these agree. And these were done on different continents with nothing conserved between them apart from the sequence and the level of supercoiling in the two. And the second one is the important one. And what we were able to see is we still see these variations in helical structure. So we see these areas where the major grooves are huge, bigger than you would expect from the crystal structure, and areas where these disappear completely. So we're seeing what we saw before on the larger plasmids. We're able to correlate this with MD simulations, which later on really helps us because they know where all the atoms are. And we see this at 2 length scales. So we're able to see this over 200 bases and 350 bases. And what we see is really nice agreement between the MD simulations and the AFM. But because we're biophysicists, not photographers, we try and quantify this to some element. And the way we did that was to use the aspect ratio, because what we've done is make a rigid planar molecule. So as you twist it or compact it, it basically gets more compacted and it gets longer and thinner. So you can think of it as old TV to cinema screens. So that's how we're classifying these. It's by the aspect ratio. And what we can see is we actually get a numerical readout of how good the agreement is between the two. And this means we can start to take information from one and give it to the other. So we're not feeding them into each other, but we're using the information from the two techniques together. And one of those is that relaxed mini circles, much like linear DNA, are very boring. They don't do anything. They don't have much life to them. They don't exhibit many dynamics. So we're in a single molecule symposium. We're talking about dynamics being important. If you have linear or relaxed DNA, it doesn't do anything. So writhe is a measure of number of turns, and this DNA is sitting very statically. But as soon as we add or take out a twist, and usually take out, so as soon as we add negative supercoiling, these structures become incredibly dynamic. They no longer sit still. They oscillate through a huge range of conformational shapes, and they move around very, very quickly. And we see exactly the same thing by AFM. So when we look at the MD simulations, what we're able to see is that the DNA is moving around, trying to minimize its number of crossings with itself, but it's missing a twist. So it is also being required to cross itself. And so what it does here is actually try and move that twist off the bottom of the DNA mini-circle over time. So it's trying to reduce the amount of crossings it has with itself. And for this, you need to remember that this is one of the most negatively charged molecules you can get. So it's incredibly negatively charged. So it doesn't want to be near itself, even with calcium ions in solution. And again, we see really, really good correlation between them. And so what happens as that DNA is moving itself around, trying to writhe itself down to its lowest energy state? Well, what happens? It seems to actually spontaneously induce defects in its own structure. And this isn't, sorry, we accidentally defrosted your plasmid, or we left it in a freezer with lots of different enzymes. We did lots of checks here. These run exactly as you would expect them to do on gels before and after. We're seeing the spontaneous formation of defects in these structures. And the way we see that by AFM, you have to follow the helix around the molecule. So because we can see this striping, what you can see is on this line, it's very vertical. And then we go around a corner and it changes by 90 degrees. And the base stacking isn't able to accommodate a change like this in helical structure. It just doesn't happen. So what that means is round that corner, our resolution has allowed us to identify that we have a defect there in structure. The other thing we're able to do, wrong button, is see areas where we have two single strands of DNA. They're much lower, they're a darker sepia. And so we move from a high double-stranded region to a low single-stranded region, and then back to a double-stranded region. And so we have these areas of multiple defects that are occurring in DNA that's not been exposed to any enzymes, just as a result of being a little bit or quite underwound, because they're quite short. And in case you didn't believe us, Agnes did a load of simulations that show that as you increase the level of supercoiling, you get more writhed structures, as you expect, so you get this compaction in structure. But at the apices, so at what you would think of as being the pin of a plectinine, the tip of a plectinine, for example, you start to see defects occurring spontaneously. And she can actually look at the level. So if we got a type 1 or a type 2, a kink or a bubble forming. And in case you don't think we can see them, we did a control where we just nicked with a BBVC1, and you can see a nice little single-stranded break. So we were able to see these, but these look very different to supercoiling induced kinks. Because at supercoiling induced kinks, what you can see is these are all happening at areas of high bending stress. So if you imagine snapping your ruler and you look away and then at some point it snaps, that's what's happening here. We're stretching the DNA beyond its tolerance, we're bending the DNA beyond its tolerance, and we're seeing the spontaneous formation of defects. And the AFM and MD correlate at what level of bending stress this can accommodate before we snap, before we give up completely. But what the MD shows us by being able to interrogate different topoisomers, different levels of negative supercoiling, is that this gets worse and worse the more supercoiling you induce, which you would expect. So from AFM, we can try and understand what's happened there. And what we see is that these defects, that these changes in structure, give us a huge difference in the free energy landscape and the number of conformations and the heterogeneity that we see. And so before supercoiling, relaxed, boring DNA, it doesn't do anything. Induce supercoiling, lots of different shapes. And we assume that that's very important in terms of recognition, because we've suddenly got a huge range of bending angles, crossings, and defects available to us. And normally in AFM, we'd just leave this here and we'd show you a picture and we'd say, thank you very much. But again, we're biophysicists and we're talking about how we can take these techniques forward. So what we've developed is an automated processing pipeline that's able to take raw data in that looks as ugly as this. That's what our normal raw data looks like. And is able to process all of our data automatically, pick out individual molecules and give us histograms of what we've got there. And this doesn't really, AFM's quite, maybe a little bit behind some of its microscopy friends like cryo and single molecule microscopy. So we don't necessarily have these tools. And this is something we're trying really hard to grow, because I think it's really where the field needs to be. It's quantifying these interactions. It's not just seeing them, it's quantifying. And so we're able to add a restriction enzyme and look at our linearized templates and say that they're the same length as our circular templates, but that we get an N of whatever. So we're able to look at the efficiency of that reaction. We're able to look at different lengths and even on imperfect data sets. And I think most people have more imperfect data sets than perfect data sets on their computer. And we're able to pick out molecules of interest within imperfect data sets. And we're able to do this over a range of lengths. And actually, if you look at the full width half maxima of those, they're only about 10 nanometers. So we get very, very good resolution as long as the samples are okay. And I have a backup slide where I can explain why we get second bad peaks on this, but it's to do with weirdnesses in annealing oligos together. And just in case you're not a DNA person, we can do this on membrane attack complexes, on supported lipid bilayers. We can do this on DNA origami rings. We can do this on nuclear pore complexes in native nuclear envelopes. So AFM is very useful for lots of different things apart from just DNA, and the stars show you where you would expect those values to be from the literature. So we see really, really nice agreement despite these tip broadening problems because we've managed to overcome them in the analysis that we did. So back to our supercoiling story. What happens when we supercoil DNA? What do these defects mean for DNA structure? Well, this is where I have to apologize to my PhD student, Carveth, who's luckily graduated and gone to work at Google and no longer have to repeat all of these experiments, because when we first did that, I assumed we'd got these wrong. Because what happens is when we look at the relaxed DNA, it's right over on the right-hand side. It's very open. It's very square, as we would expect. But as we take out supercoils, as we increase the negative supercoiling, we go right over to the left and we see compacted molecules. But if you look at the shading of these two are the wrong way around. If you think about a linear progression. So it should go lighter through to darker, and it goes light, dark, dark. And so if I show you that a different way, over here it's quite a long way away from my little eyes. Here we have the relaxed mini circles, then we take out one and we see some compaction, but then we see relaxation, and that we really weren't expecting. And this is what we redid quite a lot. And then we see that the trend continues to compaction again. And when I add these nice cherry-picked images to the top, you can kind of see why. Because what happens is when we take out one turn, we see writhe formation, which we would expect, the DNA twists over itself. But when we take out another turn, we see the spontaneous formation of defects, which opens up the structure, but in a completely different way, the circle to triangle transition, where actually the DNA now has long, rigid, straight lengths of DNA and highly curved kinks. Now, the thing to be aware of is at this length scale, here is about 0.03 to 0.06 in terms of negative supercoiling. So exactly where you'd expect interesting things to happen. So just on the left of that range, you have -1, where you have nice strong bends and rise for recognition for things that require those kind of things. And then just on the right, not very far away on a free energy landscape, you have very sharp kinks with long linear regions. So you have a piece of DNA that when it's in a small bend that you might imagine might happen at, say, a plectiny tip, you have the ability for it to buckle and form defects where you have recognition sites, which are just structural. We're not talking sequence here, where you have a strong bend, a crossing, a kink, or a linear region. So DNA is finding a way to offer its substrates anything it needs with just a very small perturbation to its level of local supercoiling. And that's quite important in recognition. And luckily, Agnes checked this using her amazing simulations. No, probably should have done that before we redid all the experiments. And what we saw again was this initial decrease in supercoiling. So we see this compaction and then the onset of defects and the relaxation of the DNA. And the fact that this is around the level of supercoiling that we'd expect to be relevant is really important in thinking about this in terms of recognition. And I think the other thing I really wanted to mention here is this work only made sense because we brought together the simulations, a lot of biochemistry, and the AFM together. I think using these biophysical techniques on their own can be very powerful, but using them in concert with other techniques can tell us so much more than each one does on its own. So in the last few minutes that I have, I wanted to talk about why this might be interesting. And to do that, I'm going to talk about when we look at G4 DNA, so fourfold DNA formed of G repeats that fold up into small G tracks. And lots of people are interested in them. They're overexpressed at oncogenes. And what we did here in this lovely paper with Ramon Villar's group at Imperial was embed these into small circles of DNA to understand, by putting small oligos of G4s into some sort of level of bending constraint, did we affect the way they fold and unfold? Did we affect their dynamics or structure? And so what we were able to see was in the absence of inhibitors or salts, we were able to see a small amount of single-stranded DNA, which I've colored in blue here. in between the double-stranded DNA that we've got in pink, we're able to still see this double helical structure, so we know what we're looking at. And then we see this little four-fold knot as a nice bright blob on this. And we see this happening repeatedly when we add things like potassium that stabilize these structures. But actually, when we zoom out a little bit and we look at a bit more of a population than one, we start to see variation in these structures. So actually this one from here to here, between these scans, which is 3 minutes, quite slow, we've actually seen this fold in real time. And so what we've been working on now is speeding up AFM, and high-speed AFM exists as a field, but speeding up AFM whilst maintaining this level of resolution. So allowing us to probe these interactions at this length scale. And so what we did, hopefully you will play, is here we have 2 molecules that we've captured at 1 frame per second. And the molecule on the left absolutely resolutely forever does nothing. Very boring, does nothing. It's unfolded. It's got a G4 there because these 2 bits of double-stranded are tethered together. And the one on the right dances like there's no tomorrow. And if you look at the bottom, it sort of kicks its legs open and closed. And as it clicks its heel together, a little white G4 blob forms there. And we think the reason that this one is so dynamic compared to this one is that actually this nick that you see in the back of these, because they're not ligated and for technical reasons, this nick that we're able to see, this darker region in the back here, allows this one, the conformational flexibility, to fold this G4 open and backwards. Whereas the one on the right is a bit more like a bow and an arrow. The single-stranded DNA is held under tension, so it's not able to fold and unfold. And so actually by looking at structures or interactions of DNA within a wider context, thinking about what it might be constrained within, and thinking about things like supercoiling, bending tension, DNA mechanics is really important when we're thinking about interactions. So I had a few more slides on looking at direct visualization of protein structure, and I'll probably just show you this one, where we're now able to do this at submolecular resolution on proteins too. So we're interested in DNA protein interactions because it's what DNA does. And so now we're looking at subdomains of proteins. So what we have here is a lovely collaboration with Alia and Chris, who are up the top. And this is looking at a small protein called NDP52, where we have 2 globular regions and a coil between them. We're able to see that singular coil, and we're actually able to measure the structure. So again, we're using these automated image analysis methods to look at the structure of proteins as well as DNA. And we get by hand measurements that relate really well with our computational methods that are able to tell the difference between the 2 sizes of these globular regions and the CTD. And we're able to look at things like dimerization. Because we're looking at individual molecules rather than a bulk population, we're able to look at these dimerizing in situ, and we're able to look at the effect on DNA. So if you want to read about this, it's on Bioarchive. So I'm going to finish there because I don't want to get us behind time this early on in the afternoon. But I'd really like to thank my collaborators, Sarah and Agnes, who are my co-corresponding authors on our Nature Comms paper and were an absolute joy to work with on this. And Andrea at Brooker, who supported me through the 4 years of insisting that it was worth making these materials. And my group at Sheffield, and I, like many others, are hiring. And I have an open-ended permanent role as an experimental officer in Sheffield. So if anyone's interested in developing microscopy techniques and would like to move to the Peak District, then please let me know.
Yeah, thank you. You know, years, I'm talking decades ago, it was predicted that dislocations in DNA would localize apices in supercoiled DNA. So it's beautiful to see it. Question I had was, so you compare different topoisomers. Obviously you've limited to unit changes in linking number when you do that. Have you tried adding more delicately, say, ethidium bromide or some sort of intercalator to almost titrate between those points in terms of superhelical stress?
No, we haven't. But I think one of the things we're currently doing in the lab, we have a 336 and a 339. And so the idea is there that we have one that wants to be in 2 different conformations because it's, and one that doesn't. So we're looking at sort of tuning it by tuning it differences in length rather than having ethidium locally in the AFM, if that makes sense. So sort of defining how happy it is to be in those different topoisomers.
Second question, really. I couldn't work out what the number, in each of your pictures you had a number up the top. So couldn't have been linking number because it wasn't integer.
Linking number, and actually it's a nasty secret of some of that, and we've since rechecked it in singles. So we added specific amounts of ethidium and purified it out rather than gel eluting to get that resolution, because even electroeluting, we can have some issues with the AFM because it's picky. So we have some of them are mixed topoisomers because it's 339, so it doesn't really want to be in one or the other. So we have some mixed populations, and I can show you how they match to a gel if you want.
Thanks. Yeah. So the question is, have you tried using this technique to measure secondary structure of mRNA? And if you have, how does it compare with the predicted structures?
So we're doing some really exciting RNA work, lots of which I can't really talk about, but I hope to be able to talk about very soon.
Next year, yeah.
Yeah, it is a great technique for looking at RNA.
Hi there, great talk. So what's the next limiting factor for the technique? And in the future, can you see the probe being improved even further?
I mean, never say never. I worked in silicon manufacturing before I did AFM stuff. I worked for Intel for a little bit, and I remember thinking 50 nanometer chips were amazing, and I think they're 5 now. So never say never with silicon manufacturing. I think we come to a pressure problem. So if we make things very, very thin, then we have to improve a lot of the resolution of our whole detection systems to compensate for the fact that we're turning a sharp probe into a needle, and we're poking biological soft objects with a needle. I think the big steps forwards here, or where we see the big steps forwards being, are in application of machine learning and better image analysis techniques to the data we have, rather than in hardware improvements, or that's at least where we're heading with this.
Thank you. That's a really fascinating talk. I was just wondering if you can potentially compare chromatinized and non-chromatinized DNA, especially how it folds.
Oh. So AFM's a surface technique, so we like to look at the top of things. So the bigger and more complicated they get, the more you're just looking at the top of them. And the more things you add in, you know, what's really nice here is I'm showing you lines and blobs in terms of DNA and protein. And the more blobs you add in, the more complex it becomes, because one of our big secrets is we don't have any chemical identification. So blobs are blobs are blobs, unless they're very size separated. So challenging, I would say.
Thank you.